found engrams at all. They've tried many times, but failed many times. The most spectacular failure was at the beginning when Carl Lashley, the great American neuroscientist, tried to find where the memories were stored in the brain by training rats and monkeys to learn new tricks, and then cutting out different bits of the brain. He found if he cut out all the brain, they couldn't do anything, but that simply proved they were paralyzed without a brain. But he could cut out large chunks of the brain, right, left, top, bottom, front, back. And these, the loss of these large brain regions didn't affect their memory. So he couldn't find memory anywhere localized in the brain. And similar experiments by a researcher called Boycott with octopuses showed the same thing. He could cut out various bits of their brain and they still remembered. And as he put it, memory seems to be both everywhere and nowhere in particular. And Lashley's student, Carl Pribram, then took Lashley's results and reformulated them in terms of his holographic theory of memory storage, that memory is stored like in a hologram all over the brain, not being localized. So the idea of being able to find a localized trace was given up as a result of that research. And so that meant that it became almost irrefutable, because if you damaged any bit of the brain and the memory still persists, you say, well, it must be somewhere else. And ever since then, people have been trying to find memories. Stephen Rose did it with day-old chicks. He taught them to respond to a moving object by imprinting, and then found that a particular bit of the brain was involved when that learning happened, labelled with radioactive glucose, that those cells were more active. They thought they'd found the memory trace. Then it turned out that if you cut that region out of the brain, they can still remember. So they just say, oh, well, they've been moved somewhere else. So over and over again since then, including very sophisticated modern research with optogenetics, where they label nerve cells with green fluorescent protein, as you know, and can actually see which cells are involved. It turns out that certain cells are involved when a memory is laid down in mice, for example, but then when they remember that memory, different cells might be involved. They only overlap to about 10% in some cases. Moreover, if you cut out those cells after a day or two, the memory is still there, and so they say it's been consolidated, it's been moved somewhere else in the brain. Yes. So whenever they think they've found it, they always end up saying it's moved somewhere else.
Yes, technically we're far beyond James and Bergson with the abilities we have in the lab to, as you say, to visualize activity in the brain as animals are learning tricks, for instance. Also to manipulate that activity, to even try to tag certain neurons and then come back to them and so on. But conceptually, we are far before Bergson and James. And yet, I would say, to put it in a way, in a positive manner, the trend seems to be moving in that direction, because now the emphasis is on networks that have dynamical properties, with technical names like manifolds, and the dynamical systems approach to neuroscience is also taking off and more widely adopted. And this leads to these brain waves going back and forth. And so this, in a way, can bridge the gap between localization of function, this natural quest to see where is that function, which is an old quest, starting with phrenology, this idea that by exploring the bumps in your head, you could tell how a person was. Well, we know that's not like that, but nevertheless, this localization of function seems to be getting distributed, dynamical, and so it seems to me that it's tending towards that really powerful idea that's still quite daunting to the sciences, and not just neuroscience. We could also speak about developmental biology and so on. So it seems we're getting there, but not as fast as we would like to.
Yes, well, what it seems to be showing, this recent research, is that it's all about wave patterns spread over the brain. And, you know, this recent work showing that normally the wave patterns would go down from the outer parts of the brain to the hippocampus, which is concerned with localization. And then during sleep, waves would go in the opposite direction from the hippocampus to the other parts of the brain in what are called sharp wave ripples. So there's this, the idea that to remember something, animals often have to sleep after they've learned it, and what's associated with that are sharp wave ripples, and which may or may not be associated with dreaming, that the memories depend on these waves, and they're distributed wave patterns. And one of the most interesting things, I think, in recent brain research is the phenomenon of representational drift, where they find when animals remember something, there's a particular pattern, a rather complex wave pattern in the brain. And when they remember it again, you see the same wave pattern, but it's not necessarily in the same place. So it can move from one part of the brain to the other. What's important is the wave pattern, not the exact cells. And so the idea that memory is stored by modifying nerve endings or changing proteins in cells that are involved in engrams, supposedly, the idea that there would be physical material memory traces localized in particular regions, is not at all supported by this. What is supported is the idea that there are wave patterns, and the wave patterns are the key thing, and those can move around. And so the brain it doesn't have to be in a particular place. So then the question comes, if memory depends on wave patterns, how are wave patterns remembered? Because to make an engram, you've got to freeze a wave pattern. You've got to freeze it and make it static to have stuff like on a flash drive or on a grooves of an acoustic record, 78 RPM or a long player. You've got or the magnetic patterns on a tape in a tape recorder. You've got to freeze the sound or the wave pattern, make it static, and then you've got to reanimate it. But how you would freeze a wave pattern and then reanimate it in another part of the brain is totally unexplained by this research.
Yes.
And it points to something else going on that's not just freezing memory traces in synapses or in proteins or in cell membranes or even in DNA.
Yes.